Good morning and good afternoon, everyone. My name is Mahalea Marshall, and I'm with the IQ Consortium Secretariat. On behalf of the IQ Consortium, I'd like to thank you for joining this morning's webinar entitled Making Bitter Medicines Better for Everyone, presented by the IQ Pediatric Working Group and sponsored by the Drug Product Leadership Group. I'll now turn things over to our speaker, Dr. Ha Nguyen from the Monell Chemical Senses Center.
So I'm Han Nguyen again. So I'm at Monell Chemical Senses Center. Today I'm talking about some of my work at Monell around beta medicines and beta blockers and how to improve the beta medicines to make it work for everyone. So that is the topic I'm talking today. So, but however, ABI. So, and then the previous study showed that the genetics or genotypes correlated with the perception of certain compounds bitterness. So here is for an example, remember that in the first study, so I mentioned that we use probe at a control drug to assess the contribution of genetics. And here is that probe is very well known for the correlation between its bitterness and the TAS2R38 genotypes. So this graph shows that the three genotypes ABI, PIV, and PIV, PIV. So people with different genotypes, they differ in the perceived bitterness of probe. So we can see here the three groups of people with three different genotypes of the TAS2R38. They differ significantly in their perceived bitterness intensity of probe. So, and another interesting data is that we know that the bitter taste perception can influenced by the genetics, and we also know that there is a data so that there is global diversity in the beta receptor genotypes. So for TAS2R38 as an example here, so we already talked about the PIV, AVI, and another rare genes AAI as well. So this graph shows the distribution of three variants in the TAS2R38 gene differs by global origins, and the three variants PIV, AVI, and AAI represent high sensitivity, low sensitivity, and medium sensitivity to the bitterness of probe and BTC, and also other compounds found in the bitter vegetable that I mentioned earlier. So, for example, compared to other ancestry, African people have a higher frequency of the gene AAI, a rare gene. So this gene represents for the medium sensitivity, the green color here. So, and some countries have a very high proportion of the high beta sensitivity gene that the orange color. You can see here some country in Asia, they almost 100% of the population have this gene, or some country here in South America. So this means that not only individual differences for genetic thing, but also ancestry or global origins might influence the bitterness of medicines, the bitterness of different vegetable. And in this case, we interest in the medications. So that's why to answer this question, whether the ancestry and the global origin influence the bitterness perception of medication and the efficacy of beta blockers, we conducted a second project. So we studied the taste of beta medicines and their modifiers in people of diverse ancestry. So in this study, we interested in different medications used to treat serious infectious disease in low-resource country, including TAF, as in the first project. Also, we have a praziquantel here for parasite treatment. We have a amlodipine for malaria. We have a TB treatments, and we also included PrOP here as a control drug. We want to see the correlation between the genetics and the genotypes and the perception of PrOP bitterness, as we know before. So for this study, we also assess the bitterness reductions by bitter reducer. So we use a bitter blockers. This is a beta blocker we used previously in another study, another project at Monell. We also included sucralose. This is a sweetener commonly used in medications and also in food and beverage to mask the bitterness. So we also included here as a control, as a comparison. And for participants, we assess the bitterness perceptions and the beta blocking efficacy across people of different ancestries, including European, African and Asian and South Asians. So we recruited participants across US and Canada. And we, with the procedure we did, we recruit participants online. We ask participants to register the study online. We sent test kit to their home, and we schedule a Zoom meeting online. And then during the Zoom meeting, we ask them to complete a sensory test similar to the first project. We ask them to taste different solutions and rate their perceived bitterness intensity on a scale, the same scale we use in the first project. And we also assess their cell-reported ancestry. We also collected DNA sample for genetics analysis. And here is the result. So as similar to the first project, the data in the first project, we observed the large individual differences in the bitterness of medications across panels, across all the medications. And we can see here the scale from 0 to 100, and across all ancestry groups, across all medication, we see that the ratings spread along the scale from 0 to 100. This means that for any of the medications here, so it may taste very bitter to some people, but not bitter at all to other people, or slightly or medium bitter for some other people. On those, the large individual differences was observed. We observe the ancestral differences too. So we see here 2 out of 5 medications differ in the bitterness by ancestry on average. So we see here prop and aminoquin. So this is this drug. So we see here European people, they perceive this drug more bitter than the other ancestry groups. So in the case of prop, so let's zoom in and look at prop bitterness rating. So then we see bitterness of prop differ among ancestries. And remember that I presented earlier, so there is global diversity in the genotypes of TAS2R38 bitter receptor across global origins. And we remember we have some countries, they have a high proportion of the sensitivity gene. So as expected, we observe the differences in the bitterness perception across ancestries. And we see here Asian people, they perceive prop more bitter than the other ancestry groups. And also, as expected, we like in the first project, we observed the bitterness of prop differ by the TAS2R38 genotype. So this is already the well-known relationship we know. And this is a way that helps us to validate our sensitive testing data. So with this, similarly, the two gene PAV, high sensitivity, low sensitivity, ABI, then we have the 3 groups of people with the different types, ABI, PIV, and PIV. So we see the significant differences between the 3 groups. So in the prop bitterness intensities. So this, if you in the sensory and consumer side, you're working on sensory and consumer data, you may know about prop taster status. So this is something similar. So the genotypes here explain most of the variations in the variation among people in the non-taster, medium taster, and super taster. So if you hear about super taster, that means this most of them have the type PIVs, like very sensitive to the bitterness of prop. We observe the differences in the bitterness of prop across ancestries. However, we did not observe the differences in the bitterness of TAF across ancestry. And we know that prop activated mostly one, almost mainly one beta taste receptors. However, in our data here at Monell, it showed that TAF is reported to activate multiple receptors, like 4 receptors at the same time. So maybe the contribution of ancestry to the bitterness of TAF is more complicated, and then we did not observe the differences between ancestries. However, we observe the bitterness suppressions differences among ancestries. So similarly, we calculated the bitter suppression score, like in the project one. The bitter suppression score here, higher number mean higher suppression, and here is lower suppressions. And the dots here represent the mean bitter suppression, and the color box here so the 95% of confidence interval. And then we see here sucralose. We have a sucralose as a sweetener, and we have a cis-methanflavone as a beta blocker. And then we see the beta suppression differ among blockers and among ancestries. Let's look at the top right corner here. Sucralose worked on Africans, work really well on Africans, almost 100% for a subgroup of participants. So it's reaching 100%. It's worked really well on African, but it's worked less for Asian people. This is interesting. So the beta suppressions for sucralose is actually differ across ancestries, and similar to sucralose, we also observe the significant differences in the beta suppression for cis-methanflavone. However, we see here the beta blocking efficiency for cis-methanflavone is less than sucralose, and it's worked for South Asian, African, but not really for European. So this is again so that not only the beta perception of medications, but also the beta suppressions differ across ancestries. So for this project, again, we found that the bitterness greatly differs from person to person, and we learned that the genetics and ancestral differences contributed to individual differences in both bitterness of medicines and beta blocker efficiency. The efficiency of beta blockers differ by ancestry. The bitterness differ by ancestry, but it's not for all medications we studied. And another outcome is that the genetic variation explains the individual differences in the bitterness of crop, but not the other medicines that may activate multiple receptors at the same time. So with that, I think that through our projects, we think that the individual differences are very important. So it helps us to enhance the generalizability and create new knowledge, and it's especially important when we want to develop formulation new products or product innovation. So to obtain broadly palatable medication or even personalized medications, formulation need to be tested on people of diverse populations. And also in the first project, we used cell-based assay to screen available or FDA approved compounds that can help us to identify potential beta blockers. So currently at Monell, we are working on beta blockers, and we also try to find the Mr. beta blocker can work on all beta receptor, can help us to reduce the bitterness of medicines as much as possible, or full suppressions as ideal, or have a universal beta blockers. So that is very helpful for medications formulations and improve the compliance, medication compliance, especially for children who takes liquid medications, and that we already know. So with that, I want to thank, this is the work in the Reed Labs. And then I want to thank my PI, Dr. Daniel Reed. She's here today with us, I think so, and all the Reed Lab members. And also we have a lot of collaborators help us to collect the data for the 2 projects. We did sensory panel in Poland, we did across US and Canada, and also many other sensory scientists involved in this project. And also Pei John here is provided with cell-based assay data and have to bring the beta blocker project forward. So, and then also the funding. So we have the funding from NIH and Monell Institution Fund and Bill and Melinda Gates Foundations. So that's I really want to thank to all people contributed to the project. So with that, I want to end my presentation and happy to take any questions.
Thank you very much for a really informative and great presentation. Do have a few questions here. Are all sweeteners equally effective at reducing bitterness? Oh, can you hear me?
Can you say it again? I can hear you.
Or, I'm sorry. Are all sweeteners equally effective at reducing bitterness?
Yeah, so yeah, that's an interesting question. Good question. So we have a sweeteners. Well, sweeteners are known for their masking effects. That is can suppress the bitterness of bitter compounds centrally in the central processing that we know about. But then also I'm working on another project regarding the beta blocking efficiency of sweeteners. So we have a hypothesis that sweeteners not only a beta masker for central effects, but also beta blocker. They may work on the beta receptor level, so that can kind of have addictive effects to reduce the bitterness of medicines. So if with that, I think different sweeteners have a different effect. So they give the effectiveness might differ across sweeteners, and we need to select the best sweeteners that work for specific bitter medications. Does that answer the question?
Yeah. Okay. Thank you. And I actually had like well, kind of a similar related question, I should say, but are you saying that, so with regard to the variability then of sweeteners, with regard as opposed to the variability of taste receptors and the response to bitter blockers, etc., is the variability look like on the one side the variability was similar? Is that true? Sweeteners versus bitter blockers?
Yeah, so I think the variations in the effectiveness, you mean the effectiveness, right? It's not
Yes.
the beta, but yeah, the effectiveness. So it depends on the specific beta blockers and sweeteners, as I mentioned. So beta blockers, so we have at least 25 beta receptors. So if we can have a beta blockers can block all the beta receptors, that's perfect. But I don't think that is yeah, that is the case. That is reality. So then beta blockers, if it's block one beta receptor, and that is the main beta receptor activate by certain compounds, then the efficiency of that beta blockers is high. And then also the variations, the individual differences mainly comes to the genetic variation or the differences in people due to that beta receptor specifically. And I think for sweetener compared to beta blocker, because sweeteners also activate the sweet taste receptors, and then the sweetness influence the bitterness reductions in, as I mentioned, the central effect. So now it's come to different factors influence the individual differences, like not only the beta taste receptor, the beta blocking the beta blocking efficiencies, the sweetness perception, but also as we know as a sensory scientist and consumer scientist, many other factors like the diets and other previous experience, many other factors influence the individual differences. So we need to look at specific medication and specific beta blockers if you want to,
Okay.
yeah, to see that.
I see. Thank you. So another question, it seemed like sucralose was much more effective at reducing bitterness for people of African versus Asian ancestry. Should formulations be tested for acceptance in different populations?
Yes, sure. So that is our suggestions. So if we want to develop, I think it depends. So if we want to develop the global, broadly, kind of universal or broadly palatable medications, we need to test on different populations and select the one that works best for all, or even not only one, maybe a mixture of blockers and sweeteners that can combine all together and work for all people across the world. Or even if you want to have a personalized medication, then you can just target a specific population, then you can improve the products. It's not only for pharmaceutical industry, but that's also true for food industry. Yeah.
Okay, thank you. One more question. Do we know which bitter receptors are stimulated by common medications known for their exceptionally bitter taste?
Yeah, so I think I'm in the bitter field for 3 years, so I'm answer this to best of my knowledge, but maybe Danny will add more. But actually that's different. So we have at least 25 bitter receptor, and we know that different medication, bitter medicines, different compounds activate different bitter receptor, and they may act different bitter receptor at the same time. But however, on those we have a 25 beta receptor, there are several commonly activated beta receptors. So for example, TAS2R14, for example, is one of the one of the top 4 activated. So if we can target 5 beta blockers that can block all the beta taste receptor, at least we can, if we look at the commonly activated, the most commonly activated beta receptor, several beta receptor, then we kind of already, I think it will work, much better. I don't know if Danny, you want to add something to this?
You're good.
Yeah.
Okay, thank you. Okay, question from the audience. Do we understand what is the mechanism of sucrose blocking bitterness?
Sucralose blocking bitterness. The sucrose or sucralose? Can you say it? Sorry, can you say it?
Sucrose.
Sucrose, right? Yeah. So as I mentioned earlier, so many sweeteners, so we know the mechanism. We kind of know the mechanism for sure that sucrose can reduce the bitterness by the central effect. So in the central processing, so it's like not on our tongues, not at the cells or the receptor level, but when the signal go downstream and go into the brain, and then the taste interactions, flavor, taste, smell, taste, flavor interactions, taste interaction, in this case sweet and bitter interactions, can suppressions, so it suppress the bitterness in that way. But the mechanism is which pathway is complicated. It's really unclear. It's not very clear yet. So, but I'm not a neuroscientist, so I'm sorry I can't tell you the mechanism that way. But what we know is the central mechanism. But sucralose that I mentioned earlier, there may be a possibility that sucralose not only work on central, but also work on the taste and the receptor cell level. So that's our hypothesis.
Thank you.
Yeah, that's why we can observe very high effectiveness for sucralose.
Right. Okay. How does the food we eat impact our taste perception of medicines despite the genetic aspect? For instance, do two people with the same genetic profile but are used to different food perceive a medicine different or similar?
This is interesting. Yeah, I don't have a clear answer for you, but I think it's a mix. So it depends on specific things. So something we know, yeah. So the previous experience and also our diets, our consumption influence our taste sensitivities as well. So genetics is just one portions, one factor, and what influence our sensitivity, taste sensitivity, or liking or preference is also comes to diet and previous experience. So I guess in that case, we have a different phenomenon, but I can give you an example like adaptations, like you consume a lot of sweet foods and the sweet solutions no longer sweet as that level as much as people consume less sweet foods, or sweet tooth people is different from the non-sweet tooth people. So I think it's, but also we had to look at specific medication, and if you care about medication and specific phenomenon, yeah. So I don't have a clear answer for that. Yeah.
That's okay. Thank you.
Yeah.
Okay.
But I don't know. Yeah. I don't know if Danny wants to add something. That's my point of view.
Okay. People in the U.S. are becoming much more concerned about additives in everything and are becoming increasingly more medicine hesitant, such as vaccine hesitancy. Is there a concern that adding sucralose to block bitterness could cause rejection of medication?
Yes. Yeah, that is something I know that people care about that, and so we have a lot of sweeteners used in food, beverages, and medicines in pharmaceutical industry. So sucralose shows the high effectiveness, but maybe another sweeteners also have a high effectiveness like sucralose. And if we can figure it out that which sweeteners can block the bitterness, which only mask the bitterness for central effects, which can have a both blocking effect and masking effect, then we can select the best option. I don't mean the sucralose. Sucralose right now is commonly used, but I don't know in the future and when it comes to the health related things. But if we understand the mechanism, and we can choose the best option, then we can choose the best option from the safe sweeteners. So that's what I think.
Right. Agree. Okay. Our last question we have here. Do you have data on reliability of in vitro tests for in vitro tests for screening new substances in means of their bitterness? And did you see types of variation in perception for your known substances and assays too?
Yeah, that's good questions. So we always collect data, retest data, so to assess the data reliability. So retest reliability. We have the retest reliability. The correlations is about 70%. So it's 0.7. That is already high. in terms of the sensory testing, because what we measure is the stage. It's not the real the stage, so it's the time that we measure. The perception is influenced by different factors. So in the second, even the first project, the reliability is a little bit lower because we did with the twins as Twins Festival, where we did central location. We set up space to test participants. They attended the festival. That's why the reliability is a little bit lower than in the second project. We did the remote testing, and we got very high reliability, around 70%. To me, based on my experience of sensory testing, that's already high reliability.
Okay, thank you very much. I think that's all the questions we have right now. So those were really great questions, and really appreciate all of your input and thoughts. Great presentation again. Thank you so much for being with us today to close the webinar.
Thank you.